So this chapter is mainly about understanding what we represent as, or how we represent habitats for microorganisms, and then how infections develop, what the outcomes are, like what are the different scenarios. Because a lot of times we may end up with number one, we have a lot of microorganisms already living in us, with us, but we also get infections, and it doesn't always mean we have the symptoms. It doesn't always mean we're gonna see the disease, but it could. And then some ideas about the origin and the transmission of microbes, and at the end, epidemiology, how they monitor disease in populations, and I gave a couple examples already today. I think we're going to get through this pretty quickly because it's less complicated. There are terms in there, and I don't think you need to know all of those. There's just some more important ones. There's some where you feel like they needed a term to summarize certain characteristics, say, but it's not something you're going to come across very often, so they're not all equally important. But I'll try to make sure that you know which ones are. So that we all know already. We have, like it or not, there's a dynamic equilibrium with microorganisms, so we do have a lot of microorganisms living with us. And just before we get into it, you can imagine that it's going to be mainly your oral cavity. It's going to be in places in your body where there's basically openings. It's not internally. You would imagine there's a lot of sterile places, but the main areas where you see microbes living are in your nose, oral cavity, up here respiratory still, and then the further you go down to the trachea, the more sterile it gets. So by here you're going to start seeing a lot less. But we'll get through where you find normal microbial growth in what parts of the body, and then how they affect you in other places if they are moved, or if they appear in other places, what that means. So I talked about the biofilms before, and the development of biofilms is important even in the natural setting. So if you think on body surfaces, you could imagine your intestines, for example. It helps to protect and stabilize these body surfaces, and it also helps to develop a host defense, the immunity that you all display. But it also causes it can also cause disease. So there's this give and take of, we need them. They have important functions, but they also can cause disease. And a lot of times the same organisms that are protecting us or living in an equilibrium that's good for us might become pathogenic if they enter the body and end up in the wrong area and the wrong places. So keep that in mind. It's not always caused by agents or by pathogens that we pick up from soil, or some that we get from other people that are sick. So your normal flora, and I talked about that a little bit a few lectures ago, how there's a trend to going toward trying to identify what our normal flora is, and it could be just like a marker. I can imagine that in a few years down the road, you might go to the doctor and they, there was the same talk about the genetic code, but there could be a simple test that shows what your main flora is, what your natural flora is composed of, and that gives them an indication of your overall health, intestinal health, and other health. And because it's seen to be very consistent in family members and generations, so your parents' flora might be pretty similar to yours, but then between us, we might have completely different ones. But what they notice is they're passed on from generation to generation, and that usually indicates that it's important. Otherwise, you wouldn't see that, or you would see a change, a greater rate of change. So what they also call the normal flora is the indigenous flora or the microbiota. Yes?
So would our flora then be genetic or more environmental?
It's a good question. I would say it's not genetic, it's environmental, and I think, for example, when you start off with breast milk, you're getting exposed to a lot of that, and so that already means you're gaining a lot from your mother, and then the people you're in contact with initially, and that's part of this lecture, is you're born and you basically go from almost sterile to a world full of microorganisms. I don't think, I mean, I don't know, but I don't think there is any kind of evidence that we would actually inherit any of those, because the microorganisms are their own entity, their own organisms, so they're not encoded in our code so that we're producing some. It should be environmental, but because we're exposed to, especially initially, to the same environment and to our parents' microorganisms, we probably pick up most of that. But if it wouldn't be important, you could imagine that you're gonna that's changing over lifetime. It would be interesting with, for example, you do twin studies or adopted children studies and see how their flora compares to that of their natural parents, and then to that of their or their biological, and then their parents now. So I imagine it's environmental, the conditions you grow up with. And then infection, and that's always important to, a lot of times disease and infection is used in a similar sense, but really what infection just means, the microbe is actually penetrating the host, gets through the defense and multiplies, but the disease means what actually the damage that this action has, and sometimes you have an infection but you won't see the disease. So infection isn't equal to disease. Disease really means you find, and we'll talk about that too a lot more today, damage and disruption of tissue and organs. So make sure you know that you make the distinction between infection and the disease. So those would be this is the infection can lead to the infectious disease, and this is the unfortunate outcome, I guess, and then you have the normal flora. So that's just showing that it can go either way. There's like this equilibrium of normal flora versus infection. And then we don't have to go into details here, but for example, Streptococcus pneumoniae lives in the higher parts of your respiratory system, and it's pretty normal. But it also can become, it can also be an infectious agent and become a problem, and it depends on where it moves or where it's introduced, what area it's introduced to. But in general, you have these specific areas where you always find a lot more microbes than anywhere else in your body. And you would say in this case, you would see Streptococcus pneumoniae in these areas, maybe up to the trachea here somewhere. But then if it would actually enter into the sinuses or down to the alveoles, that's where you have sterile tissue that shouldn't be exposed to it, and that's where those that Streptococcus that's part of your normal flora would create problems. So here what they do is the microbe, this is the action of microbe, and the beneficial effect would be it colonizes, and it is kept in check. There's immunity, but what can also happen if it crosses the line of defense and enters sterile tissue further down or up here. Meningitis is one of the disease that can be caused by infections here. Then that can become a problem. And so once they cross that line and get into sterile tissues and multiply there, then it can either be caused by the immune system, or it can actually affect the tissue and then lead to disease, which can lead to damage of organs, can have all kinds of consequences. So most areas of the body that are in contact with the outside environment are obviously places where you can find microbes, because the contact with the outside is where you have constant infiltration or a constant exposure to microbes, and so you find those there. And then the internal organs, tissues, and fluids are microbe-free. Then you have some transient microbes that are only there for a short amount of time, and then you have residents, which, in those areas you usually have residents where a certain your microbial flora would be composed of residents. A lot of them are established in it. So skin, obviously we've talked about that. We've taken samples from the skin, took some samples from the scalp. Upper respiratory tract, the GI tract, external genitalia, outer opening of the urethra, vagina, external ear and canal, eye. Those are the places you find a lot of microbes. I'll give you a number. Later. But the interesting thing is some of it has to do with your personal hygiene, and some doesn't. So that's another interesting side note. But I'll get to that. So we talked about these super infections already, and the bacterial flora, the normal flora, benefits the host by preventing overgrowth of harmful microbes. So you can imagine you ingest a harmful microbe, it gets into this established area, for example, in your GI tract, and it's facing competition. Whereas if that would be a sterile place, it would be much easier for that microbe in terms of the supplies, in terms of the substrate and everything, if it's in the right environment, to reproduce fast. So there wouldn't be any microbes producing antibiotics, for example, things like that. And this is the infection I was talking about before, endogenous infections, where you see microbes or microorganisms that are part of your normal flora, and they're introduced to a site that was previously sterile. So that's an infection that's actually happening through a microorganism that is part of your normal flora. So the initial colonization of the human is usually done when the fetal membrane is exposed. So when through the actual process of birth, and after that, there's a lot of handling and feeding throughout the first. Throughout the first hours, but also throughout the first months, days, months, years, and so you would continue to establish that flora. And we talked about how newborns shouldn't be eating honey that's completely perfect for us, because the flora is not established enough to deal with certain microorganisms you might be introducing with it. There are some exceptions. I think that might come next, or a little bit later. So there's some exceptions. That there are some microorganisms that can actually pass through the placenta and then make their way from the bloodstream of the mother to the baby. So there are some infections that can actually cause damage in the unborn fetus because of the ability of some microorganisms to pass that barrier. And I think there's a couple examples later. This one. And so what I said earlier is, if you think of the human skin, so we'll just look at those few areas where you do have a lot of microorganisms, and we'll start with the skin because it's the largest area, it's the most accessible organ, and you have usually two populations, a transient one and a resident one. And the transient one, and that's where the hygiene comes into place, is mostly on the epidermis. So you have cracks, you have pores, etc., etc. So you have microorganisms living up there, and they are that depending on your hygiene. You've probably seen people where you imagine they have a lot of microorganisms on the top layer. I have. But then you also have to remember that a lot of this first layer, your skin layer, has deeper sections where the hair is based, and there are glands and pores. So there are bacteria living in here that with your regular hygiene you wouldn't be able to eliminate. And down here shows skin with some rods and some coccypes. They're red, so you can't see them, but it's amazing. I mean, number one, if you would really see what is that, maybe a 10,000-fold magnified image of your skin, I mean, what's going on there? It would be pretty interesting to do that and scan your body, but not everybody has an electron microscope at home. One of the main other areas is the GI tract, or gastrointestinal tract, and it makes sense if you look at the development of I guess they call them deuterostomes, but mainly how we develop from a zygote, from one cell to a human, or a lot of animals, is we have this tube going from top to bottom, and so that tube basically has two openings, and you're constantly, number one is you have openings here, so you expect bacteria here, you expect bacteria at the end, at the exit, but you also have to know that you're constantly taking in food. So you're constantly, none of that is sterile, so you're constantly exposing your intestines to microorganisms. Where would you, so what you have to distinguish though is you find a lot more up here, so in the oral cavity down to maybe here, and then further down you see less. And then the stomach is an exception because, or not an exception, but you have, because of the stomach acid, already a much more limited number of microorganisms, because the acidic environment doesn't allow a lot of microorganisms to do well here. And then, so this area from up here, somewhere in the respiratory tract, higher up down to the large intestines, is actually a lot, or there's a lot less in terms of normal flora. And then the large intestines, and then the rectum, there's a appreciable flora. So there's a lot of there are a lot of microorganisms, and here those are the ones that we a lot of times need for helping to digest food, etc. So the mouth actually has the most diverse and unique flora. So there's in the oral cavities where you find the most species. They say they've identified about 600 species that live in your oral cavity, and per millimeter of, I guess, saliva, they find 5 times 10 to the 9 cells. So that's per milliliter. So this is what's going on in your mouth. In case you're thinking about kissing somebody foreign. And the other thing is, what's also important is, that's the count of the saliva actually. And what's also important is that they were saying, this is the production of the saliva is a constant process. So you're producing it, and it's quickly, it's I guess from the gland on, you would assume that's sterile, so you acquire those quickly because of our exposure to air and all kinds of things, food, water. But what they say is, if you think about how well a mouthwash really works, if you're constantly producing saliva, and that's the concentration of cells, they can imagine that you take it out and yes?
But isn't your saliva sterile once it comes out of the gland if your mouth is dirty?
Yeah.
So if you're producing the saliva and your mouth is clean, then
it might change in terms. Yeah, it might change, but you can imagine from just our experiments how long it would take for your mouth to be dirty again. Yeah. It probably is a good idea. I'm not anti mouthwash, but at the same time it tells you how if you for the nighttime, for example, make sure you close your mouth and don't breathe in, breathe through your nose. Well, actually, that goes in there too. So I don't know how to prevent it from having maybe a mask, and then in the morning. But you should also embrace the microorganism and say it's happening to all of us. So it's not like it affects us, negatively. And who wants to live that long? I don't. So the large intestines, you can imagine that a lot of that happens obviously towards the end of your digestion, and the estimate is that 10 to 30% of your fecal volume is just microbes. So that's a huge amount. There are really important interactions with the host happening in the intestines. They favor anaerobic bacteria, and it's clear that that's what creates a lot of the odors, a lot of the gases. They come from bacteria in the intestines. And I don't know if there's a number, but what I hear, maybe 8 liters of gas or something is the production. It's amazing, like how much gas is actually produced in your large intestine. And I don't know what happens with it, most of it. Really, there's only a small percentage that you can actually get rid of. So I don't know if it gets reabsorbed, if there are other organisms that use it, but there's a huge amount. I mean, if you think of like 8 liters of gas volume, that's why you can see. I mean, I don't know if it's in the book too. I think I read it in this book. There are explosions when there have been explosions. No, intestinal surgery has caused that before. So there was just, and it damaged the large intestines. You'd have to look that up. I didn't want to go further into the matter. But it's pretty, see, he doesn't say shut up to me.
She was saying it to Jim.
I know, that's what I'm saying. So those are the two: oral cavity, gastrointestinal, and then, no, skin was the first one. So respiratory tract, oral cavity is what we're looking at next. And so you can imagine, since it's got such a broad spectrum of microorganisms, they're basically living in this area all the way down to here. But around here, the concentration declines significantly. So it's mainly from what comes in through your airways. And there's some examples. Streptococcus in your oral cavity, nasal entrance. You got aureus in there, which we've worked with, I think, or at least tried to. Sometimes we don't get what we're supposed to get. Some not sure, but I know we did with this one. And we didn't we also get that from? I thought we got that from somebody's.
From our skin.
Yeah, did we try to get it from our skin? So there are these two mucous membranes, Neisseria, and then you've got some other microorganisms in tonsil and lower pharynx. So in this area, so different organisms. Yes.
Is that where strep throat comes from? Streptococci?
Possible. I don't know the origin. Could be. Would make sense. You have to look that up too.
Always homework.
So another one is the genitourinary tract that we mentioned before, and there's microflora. You got the outer opening of the urethra in both cases, both male and female. Vagina is one. In addition, and interesting here is for females. I think the theory is that estrogen changes the composition of your microbial flora, meaning before or through puberty, you start developing through this estrogen. You start developing a more of an acidic environment, and that actually lasts as long as you're fertile, because it's supposed to, this lower pH is supposed to protect the fetus. So before puberty, and then after or menopause, you see these changes in between that actually change through glycogen secretion, fermentation, and a lower pH create this pH that's supposed to protect the fetus if you're pregnant from pathogenic microorganisms entering the environment of the fetus. At least that's, I don't know if that's completely proven, but that's the theory behind it. So there are changes to that not here. There are changes to this. No, there are changes to this flora. I mix up the male and female reproductive system all the time. So hard to remember. So we already know this about the normal resident flora. So it's essential for our health, and it prevents infections, enhances our defense, because we're having, the immune system can actually practice on microbes in there, and it's altered by a lot of things like antibiotics, taken by antibiotics, by changes in our diet, disease. So it's not a given that you have the same flora all the time. And then we've encountered I don't know how it is now, but there's definitely a more of a trend to sell probiotics to support your natural flora. And then I was writing down there, what else have you heard of? What else can you do if your natural flora is destroyed? You must have heard about something. Your natural flora is destroyed. How can you
Lily
don't go
blocks
there?
that out. Doesn't chemotherapy do that? And it destroys everything.
Oh, you mean, so I was thinking more of chemotherapy. Well, a lot of it definitely suppresses your immune system, and then also a lot of the microbial. But it depends on. It's just like any other chemicals. It depends really on how. Or it depends on the microorganism that's affected or not. I don't know if it would, you wouldn't probably be able to survive a complete knockout of all your microorganisms. But, so everybody remember what was it called, fecal, what was the second word? Thanks, transplant. That's what I was referring to. So, not that I want you to focus on this, but it's another way of saying, another method of and people, there's lots of evidence that it's actually really good. It's another way of getting your natural flora back on track if you have lost it for whatever reason.
Couldn't they do the same thing with the stomach, flora from the stomach rather than from the colon?
Well, if you remember the earlier picture, there's not much in the stomach, so you would have to basically, you would transfer but the other method was, I should have referred to that, is the synthetically produced soup, right? That's what they call it, the soup. So if you think of like what tissues are sterile and what aren't, what would you put in here?
All together.
Did you? I thought I heard. But you said lungs, right?
Right.
Okay, that's the right answer. So all these are exposed mouth, hair follicles. It's a little bit deeper, but still exposed to the external pharynx and lungs.
If stomach were there, that would be the same.
I think. Yeah, that would be, probably part of, not completely absent, but it would, that's why it wouldn't be in there, because it would be confusing. But lungs are really, I remember hearing about a friend's dad who got a lung transplant, and the biggest problem, and that's just, that's just how I think about things sometimes, to make them easier to understand, but the biggest problem with a lung transplant is that you're basically, just like with any other organ, if you receive it from somebody, you have to immunosuppress your body for the whole entire time you live, because it is always going to try to reject that. It sees it's not yours and it wants to reject it just like anything else. So your immune system has to be kept really low, and then your lungs are the biggest problem in this case because there is exposure to air all the time. So then if you get these particles in, and some of them make it down to the lungs, you're basically immunosuppressing an organ that also needs the help of the immune system. So a heart, for example, it's in a sterile spot, and you would imagine that if you transplant that, you immunosuppress, but it's also not exposed to as much. But the lungs, there shouldn't be as much, but it's clearly breathing air in and out, there's a higher chance that you get infections in the lungs. So it's a very difficult, it's a really, I don't know what kind of disease creates problems that you actually have to get transplants, but that's on a list of transplants, that's probably the last one you would want to pick because of its exposure to the environment. So now that was more of the natural flora you have in your body, but then there's also the possibility of an infection. And so there's a portal of entry. How does the microorganism actually evade the barriers, get in, and then and we already know a little bit about that. You'll find out, and then it actually has to leave the host or can leave the host, which is important for us because if it leaves the host and it's a way of transmitting a disease, then you would obviously want to know what that portal of exit is. And then we're also going to look at what happens in your body. How does it go from actually entering to multiplying to actually affecting certain tissues and damaging those. So this is just a short list, and it's clear to everybody if I ask you to name a couple factors that would increase your susceptibility to disease or to an infection. You would probably say, well, age is probably one, or stress. Everybody knows that if you're too stressed, that's what I tell you. No, that's the thing you aren't. That's the one thing you aren't.
How do you know?
Just kidding. You just make that impression.
You know me.
I might act like I'm not sure.
Are you just counting?
Oh. What am I doing here? No, I'm counting so I know how many scripts.
Oh, okay.
There's people.
I thought that there's about 4. Are there people missing?
Oh, yeah.
Or 5?
So 23 is usually the total. Yeah. So those are all, you can read through them. It's clear that those are all part of, or those are all factors increasing the susceptibility to infection. And what you should distinguish between are true pathogens and opportunistic pathogens. So there's some that are capable of causing disease in healthy people. And if you think about last year's in the winter, did anybody get the flu last winter or last Christmas time? Yeah, you did? No? Did anybody get a flu shot? Yeah?
Yeah, I did.
Hell,
I know.
every year. Injecting your body with the flu.
Every year.
Yeah? Well, the thing is, I was asking about that too. Do you think, and I don't know that much about it, do you think the flu got more, the consequences or the consequences of the flu are more severe than they used to be? Because, 20, 30 years ago, nobody knew about a flu shot. It was like, you get the flu, you make it through. Is it more convenient, or do you think, because I don't know really.
I think there's been more super strains that have come through, like the H1N1.
Yeah, bird flu and swine flu.
And
I
also
think.
just more communication. You hear about
Yeah.
people dying from it more than you used to.
Yeah. I know we had to, we were traveling, and I heard, there was a Christmas time, and the funny thing was that I think they said at the hospitals we had to frequent, that this year's flu shot was not very accurate in terms of what it covered.
There are a lot of people still getting it even after the shot.
Yeah.
Yeah. But they have to guess in like a day or
There's so
something.
many
Right, that's what happens. Yeah.
No, no. Oh, okay. Yeah. So you think that, because I was wondering if they initiate it, so they base it on what they find initially. Yeah. Okay. So anybody actually, any doctor, and I've seen some conservative doctors, they would still tell you to do it.
They're making all the doctors do it now,
Yeah.
too. They won't let them come in the hospital.
And is it because of the cost, or,
You're a medical worker to have them.
Yeah.
I'm just wondering, is it, you know, what's the, is it the, you know, in terms of the interest of, you know, the government, you know, the Center of Disease Control, is their interest just preventing this huge amount of all the problems of having the flu, or I mean, because I feel like, you know, like I said, it used to be just something you had to go through, and there was nothing
Really?
really.
I remember getting flu shots as a little kid.
Really? You guys grew up here. I noticed that German was always underdeveloped.
Yeah, right.
No?
Hear about all these germs.
So you got them every year, pretty much, huh?
Yeah.
You got a lot of
That's
shots.
awesome.
That's crazy. Yeah, because I haven't gotten one yet.
You haven't gotten a flu shot?
No, but that was
Why?
this last year was the first year where I thought I should. Well, I don't know. I was just not even aware of. I should see him driving by a CVS or whatever. I think, wow, yeah, it says flu shot, and I usually don't get sick, and then now with kids, I feel like I should really get one.
Yeah, and especially where you work, too. Like, I've always worked in restaurants.
Yeah.
So it's like, they
Yeah.
get it.
I always work with sterile students who spray their tables even for lecture. So I'm in a low risk group. So the opportunistic pathogens are the ones that actually affect you or can cause disease when your immune system is compromised. So in a normal one, the plague, malaria, influenza, healthy person, you get infected, or you get that you're exposed to the pathogen, you get sick. These ones are more for some, like the Pseudomonas I showed earlier, or yeast infections, where you say if there is some immunosuppression going on, if there's the body's not completely able to function perfectly, that's when they become pathogens. And what's important is it depends a lot on the virulence of the pathogen. So there's a lot of different characteristics.
Virulence again?
Well, the virulence is for the factors are virulence factors are factors that increase the possibility that the disease is more severe. So there could be, you know, one of it would be, you know, what the host is like, what actual tissue the pathogen is in, things like that. So higher virulence means more severe, or a more virulent pathogen means usually the disease is more severe. And then what's important, obviously, and that's actually one of the questions you know, a problem I created with that question in the lab test when I asked you why you know, what's the difference between our tables being just not really, you know, treated as much as the treated as, what I meant with that question is not the plates we're getting to work with, but the plates we're done with. So we're experimenting with microbes, and then everything goes into the red bucket and goes into the gets completely sterilized. How come we're working here with the stuff? And I got a lot of good answers of people saying, well, we're not actually inoculating the table. It made me laugh, but it's true. We're not. You know, the concentration is what all I wanted to hear is the concentration difference. So the concentration in plates is high. The concentration in our environment is low, and so that relates to the infectious dose. So the dose is always anything you do. You know, wasn't there somebody doing years ago a bet how much water they can drink and die from it or something? Wasn't that was a dose, a concentration question. So you can tell how much or how I mean that was an insane dose or insane amount of water, but in general that makes the difference. So if we have a high concentration of organisms in the plates, because we try to grow them, we need to sterilize it, whereas here, the environment is probably not worse than in your regular public bathroom. So if the infectious dose is smaller, those microbes have greater virulence. So again, it makes sense with a little dose of this microorganism, you get the same effect. Means you have the organism has a greater virulence, and then the infectious dose really is the minimum number of microbes required for an infection to proceed. So if you're lower, if it's a lower dose and it's kept in check, there's not really anything happening. So that makes a big difference in terms of where this infection with this specific microorganism ends up, or how far it can go. So a lack of reaching the infectious dose will usually not create an infection. So in some cases you really need nothing. Like for measles, you need one virus particle. That's the infectious dose. You're exposed to that. You depending on it says generally. So you might depending on your overall if you're for example had it already, or if you have antibodies for it, etc., that's a different story. But in general, you can determine here what the difference is, how much you need. You need 10 to 100 cells in some cases. You need up to 100 million cells for cholera, and that has to be ingested. So that means for cholera you really need a huge infectious dose, and so that kind of gives you an idea of the conditions that have to be in place to get infected with cholera, because you need such a high concentration. So it starts all with the entry of the microorganisms, how they get established, and so they call them the portals of entry. And obviously those are what we've just talked about, the skin, and what it is a lot of times, and that's especially for nurses. I mean, they talk about that a lot. Punctures, needle punctures are really a huge problem, because you handle so much of that it could be a small, and there's a case study in the book where some nurse just at the end of the day gets a needle stick, and then had to go through all these tests because the patient has had a HIV, and then it turns out he also had tuberculosis, I think, or something else. So it's amazing. You think it's just one little needle stick, and you think, I mean, I would not be able to work in this environment, because I know I would be like, and then done, go home, not say anything. Yes?
Yeah, my stepmom was working for an AIDS and hepatitis C clinic as a RN, and she contracted hepatitis C, which put yourself on.
Yeah, it's a huge concern for sure, and that's not another thing. There's a few slides on just what goes on in a hospital and how they try to control it. So they usually have health management, where you have a person or multiple people to just constantly try to control the exposure to patients, in any sort and form. So handling fluids, handling contact, everything, and it makes a lot of sense, because it's really you think hospital always helps, but it concentrates all the sick people. So it's a place where you have to be. It's where it has to be really important to be absolutely to make that decision. Now, are you going to be able to follow all these rules? No.
Yes, I would. I'll be stressed out about it all the time.
Did I have to say that?
I'm out of here.
See ya. Going back to that.
So that's for skin. The GI tract, foods, water, drink, anything ingested. That's why they tell you keep your hands out of your mouth, keep your fingers out of the mouth. That's what you tell your kids. It makes a difference. Respiratory tract, same thing. Keep your hands out of out of your nose. And then for urogenital tract, there's entrance through sexual contact, displaced organisms. So there's and then there's what I said before, placental. So through the placenta, there's some areas or some organisms that are actually able to travel from the mother's bloodstream into that of the fetus. And so here they're just showing some of the main areas. But it's clear that where you have all the microbes, naturally is where you also see them entering your body. And in addition, you have these, you have injuries to your skin, for example, that create those. And usually, if you have tetanus, so for example, you have prevention in place for certain infections, because if I get a rusty nail into my skin, or if I injure myself, there's a high likelihood that you're gonna be exposed to some of those bacteria, and then you want to be prepared for it because they're really, really toxic. So here's what I've talked about before, and what they you can memorize a few of them. What they usually write down is STORCH, storch, for the different. So syphilis is one of them. I was remembering for the different microorganisms that can actually penetrate or pass the barrier of the placenta. And so here in this area where you have the maternal blood vessels are over here, you can actually, these bacterial cells can actually make it, and then through the umbilical cord enter the bloodstream of the fetus. And so those are some of the disease you could be born with, because you've had these basically they were passed on to you from your mother. And then this one, that was just one of the areas of entry into a body. So that would be one of the earliest examples of getting exposed to microorganisms in the unborn. And then in general, if you look at all the other areas where we find microbes, what they need usually is attached to the host in some way. And we learned about these structures already. And so you have these, receptors a lot of times for viruses. We talked about those and how they are able to actually interact with the host cell. So that would be for viruses. You also find these kind of structures. Where would you expect those? Suckers, hooks, barbs, worms. So you have parasitic worms. That's how they actually attach to tissues, and then can reproduce and live in the host by those means. And then you have, for bacteria, you have a lot of those. You have Cilia fimbriae that allows you. Glycocalyx, remember we had this slime layer that was able to kind of glue on. So those are important ways of attaching to the host cell. And then once the microorganism has entered the host, it depends on those virulence factors, how severe, how serious the disease is. So it determines the degree of tissue damage that occurs, the severity of the disease. And there's a couple things that can happen. So there could be a production of exoenzymes, and we've talked about those before. Remember endoenzymes and exoenzymes and where they work. And that was probably part of the chapter test too. So and it's indicated in their name, they're given off or produced by microorganisms and then leave the microorganism or the cell, and they're able to, for example, dissolve extracellular barriers and then actually make it through certain cell barriers. For example, you've got here cells, epithelial cells, and the bacteria up here are able to infiltrate this tissue, and then after the exoenzymes have broken this barrier. So that's one of the ways. And then toxigenicity is the possibility of, or the ability of a bacteria to produce toxins, exotoxins that then break the cells. So that's another way of entering and causing damage. So you would have bacteria on the outside of the cell layer producing toxins that actually destroy these cells, and then bacteria are able to infiltrate the underlying tissue or bloodstream, any kind of fluid. There's one other way, I think. There's one other possibility. Some pathogens actually produce a specific, or they secrete a compound that allows them to actually become part of, or to nestle into a cell and then be internalized by the cell. So that's just a specific one way of entering the body. And Salmonella is an example where there's first adhesion by the fimbriae, and then a protein is released, and it disrupts some of the structural actin here. The cell can actually sit in this, what they call a pedestal, and then it's pulled into the cell, and then it can reproduce internally and then move further on in tissue. So it's just one of the pathways. This specific microorganism can make it into the tissue. And then what's important is how do those cells survive the host defense? If they make it in, there's usually automatically a defense in place that recognizes the microorganism as foreign. And it's just like with foreign organs, foreign objects, your body usually reacts immediately to foreign. And then we'll probably go through some of that in immune system immunology to discuss what those how it's recognized, how it recognizes own and foreign. But a lot of the initial response comes from phagocytes, so cells that actually eat up or internalize and digest the cells or the foreign cells. And so some of the microorganisms have anti-phagocytic factors, so factors that work against those phagocytes and avoid those. So an example would be Staphylococcus and Streptococcus producing leukocytins, which are leukocytes, white blood cells. So they're actually producing toxins to white blood cells. And then we had a question on this, the slime layer and the capsule, and how that makes the phagocytosis difficult. So it's an additional, it helps attaching, but it also makes it, because it's a strong layer, it helps it avoid phagocytosis. And then there's some which is interesting that are actually not fighting it, but some of them are actually able to survive in the cell. So they actually are internalized by a phagocyte, and then they're able, in this case it's a different example, but they're able to live in the phagocyte, and that's a problem because then you have a microorganism in a cell that you call your own. So the immune system is not looking for it anymore. So they live in this phagocyte and can actually reproduce in there and live in it. So that could be a disease that you see much later. So the outbreak doesn't initially happen because they're actually internalized and stay alive in those cells. So when it comes to the damage that the bacteria can cause to our organs and tissues, we already talked about the two toxins, endotoxin and exotoxin. And so those are usually the exotoxins secreted by the bacteria, and they have usually they're specific for target cells. For example, hemolysins target red blood cells. So there's a toxin that targets red blood cells. And there are also, which I'm going to show a little bit more, there are AB toxins, where the toxin is actually able to bind with a B side base or whatever with a specific structure, and then the enzyme, and then you have an active part of the toxin. So we're going to look into that a little bit further down, but that is part of how botulism works, through a toxin that has a binding site to the area where you have the acetylcholine neurotransmitter go through that neuromuscular gap, and then the toxin that actually, or the active part that actually blocks it. And then endotoxins, they're usually they're not secreted, so they're not secreted by the microorganism, but released after the cell is damaged. So it's a more general effect. They're not as specific. It's a more general effect on your whole body. And this is what they would look like. So you would have these exotoxins that are released constantly by the bacteria and affect different target organs, heart, muscle, blood cells, and then the endotoxins that are actually releasing the toxins when they lyse, and that's where you have like a more general effect. It's less specific, it's more of a general physiological effect, fever.
It looks like it should be the other way around, like endotoxins, they should be within the cell.
Well, these are they're in the cell, and I think this shows after the cell lies. So the cells are breaking apart and the toxins are out. So if you compare those, and I don't worry about knowing any of this. It's just they're obviously here specific versus more general systemic effects. So systemic meaning your whole body shows the effects: fever, inflammation. I don't know if there's anything. So releases, they're not secreted from the live cell, but from a cell while during lysis. That's pretty much all we need to really know between the two. And this is how the AB exotoxin works. Like I said, there's an active and a binding component. And so the active one is an inhibitor, versus the B1 binds. So in botulism, you would imagine that the binding site has some way of binding to the gap between the nerve and the muscle cell, and then the active site, the active part of the active component of the toxin is actually causing the effect in the neuromuscular synapse. So one binds, the other one has the actual or does the actual inhibition and the damage. So if you go through infection and disease, so say we've gone through your regular, you have your microfauna or the fauna established by microorganisms. That's normal, and then you have and that's in specific areas in your body, and then in some, if you want to look at pathogens and see how they enter the body, there's different ways, and you're constantly exposed to air, to food, to water, and so there's specific areas in your body where you find more microbes, and then they're able to enter your body through specific mechanisms like exoenzymes, and then they can produce toxins that damage your tissue. If you have acquired, or if you are